Antibiotic resistance is fast becoming one of the biggest challenges in modern medicine. Basically, bacteria are getting smarter. They're evolving to survive the very treatment that used to kill them. This means that infections that were once easy to cure are now becoming harder and sometimes even impossible to treat. In this video, we'll look at how scientists study this problem using something called the Kirby-Bauer test, which measures how effective different antibiotics and even natural essential oils are at stopping bacterial growth. We tested things like tea tree and eucalyptus oil alongside regular antibiotics on both gram-positive and gram-negative bacteria to see which worked best. By the end, you'll see how natural compounds might play a surprising role in fighting bacteria, and why understanding antibiotic resistance is so important to protect our future health. As mentioned, one way to test antibiotic resistance is using the Kirby-Bauer test. Here's how it works. Small discs soaked with antibiotics are placed on a plate covered in bacteria. After some time, you'll notice clear circles around some of the discs. That's called the zone of inhibition. It shows where bacteria couldn't grow because the antibiotic was effective. If the clear zone is less than 6 millimeters, the antibiotic doesn't really work. Between 6 and 60 millimeters, it means that it was somewhat effective. And anything greater than 60 millimeters means that it was highly antibacterial. So in short, this test gives us a visual way to see which antibiotics or even natural substances can truly fight off bacteria.
The materials to conduct this experiment include 1 gram-positive bacteria, 1 gram-negative bacteria, 1 micropipette, 1 box of micropipette tips, 2 broth plates, 16 filtered paper disc, 1 pair of forceps, 1 bottle of ethanol, 1 ethanol lamp, 1 cell spreader, 1 permanent marker, 1 vial of essential oils, and 1 dispenser of each antibiotic. Here are our procedures. 1. Divide the agar side of the LB agar plate to create 7 equal sections. 2. Label the sections 1 through 7 near the center of the agar plate to avoid covering bacterial growth. 3. Inoculate the LB agar plate with 130 microliters of bacterial culture. Replace the lid and discard the pipette tip. 4. Use a cell spreader to spread the bacteria evenly onto the agar surface. 5. Cover the plate and let it sit at room temperature for roughly 10 minutes so it can absorb. 6. Sterilize the forceps and wait 30 seconds for the forceps to cool. 7. With the sterilized forceps, place treatment disc near the outer edge of their agar plate. 8. After placing the lid on your LB agar plate, for the last time, allow it to rest. The agar needs to be upside down, undisturbed for 5 minutes. 9. Incubate the plate for around 37 Celsius and for 24 hours.
The results indicate that ampicillin and tetracycline were the most effective agents, particularly against gram-positive bacteria. Tea tree oil and eucalyptus oil also exhibited antibacterial activity, though their effects were notably weaker than those of the antibiotics. Streptomycin demonstrated limited efficiency, while almond oil and water showed no inhibitory activity. The results indicate that both antibiotics and certain essential oils can inhibit bacterial growth. Ampicillin and tetracycline demonstrated the highest overall effectiveness, while tea tree oil and eucalyptus oil also exhibited notable antibacterial activity. This suggests that natural compounds may serve as potential alternatives to conventional antibiotics. Streptomycin showed comparatively weaker effects, and almond oil exhibited no measurable inhibition. Overall, the findings were consistent with expectations, although the strong antibacterial activity of the essential oils was unexpectedly pronounced. A possible source of error could be that the concentration of the essential oils varied between discs since they were dipped and then dried on paper, which may have caused uneven absorption. This variation could have affected how much active compound was applied, influencing the size of inhibition zones.
One major way to strengthen this experiment would be to use a larger variety of both essential oils and antibiotics. This would allow us to collect more comprehensive data and make more accurate comparisons between treatments. Another improvement would be to expand the range of substances tested, which would improve the reliability of the results and help us identify clearer patterns. Lastly, repeating the experiment multiple times would be essential to confirm consistency and reduce the effects of any possible experimental errors. This would make the data much more dependable and scientifically valid. A possible source of error could be that the concentration of the essential oils varied between discs since they were dipped and then dried on paper, which may have caused uneven absorption. This variation could have affected how much active compound was applied, influencing the size of inhibition zones. A key next step would be to explore combining essential oils with standard antibiotics to see if these combinations could increase the zones of inhibition. By doing so, we might discover ways to enhance antimicrobial effectiveness and even reduce the dosage of antibiotics needed while still maintaining strong antibacterial effects. On a larger scale, this line of research could lead to the development of new treatments or medicines capable of targeting specific diseases or bacterial infections. It would also be valuable to examine how different types of bacteria, both gram-positive and gram-negative, respond to various natural and synthetic antimicrobial agents. Finally, scientific communication is the process of sharing scientific findings both within and beyond the scientific community. Its main goal is to make knowledge accessible and understandable to everyone. Effective communication is essential because it helps people make informed decisions that can improve their health, safety, and overall well-being. To be effective, scientific communication must be credible and easy to understand. Some key necessities when communicating science to the general public include being clear, concise, and engaging, helping audiences identify reliable sources of information, and avoiding complicated jargon by using simple, everyday language.